Hi, my name is Aidan Draper and this video is about my summer research project here at the University of Notre Dame where I used light reflectance data to classify marshland grasses from space. I spent my summer working in the McLaughlin lab with this guy, Dr. Jason McLaughlin, but also with these guys, saltwater marshland grasses. These grasses are part of an ongoing research topic in the McLaughlin lab and the Smithsonian Environmental Research Center, which owns a special piece of marshland located here in the Chesapeake Bay near the Environmental Research Center. For years, they have been researching the influence of rising CO2 levels on different species of grasses in their marsh. This octagon here is actually pumping CO2 directly into the air surrounding some of these plants. This is in order to see how plants might react differently based on the CO2 in the atmosphere. Additionally, the lab and the research center have seen a spike in the abundance of an invasive species called Phragmites, which may also be disrupting the marshland ecosystems along the eastern U.S. coast. For my analysis, we were kindly given a dataset of 514 1x1 meter plots within the research center's marshland. These 1x1 meter plots were surveyed every 20 meters. Each row in the dataset had ordinal rankings indicating how much of each of the 6 grass species was visually observed in the 1x1 meter space. This data would be used for the training set in my project. This brings us to our investigation. We are curious whether or not we could use Landsat satellite photos to classify grasses in our marshland from space. We had some intuition that we would be able to do this because remote sensing experts have used the random forest model to classify different areas of land observed from space, like agricultural plots, urban spaces, bodies of water, and forests, because of their greatly differing light bands. However, the random forest model has been used far less to classify spaces that just slightly differ in their light reflectance bands. Like you might see here with different grass species, these bands are far more difficult to differentiate than the previous bands. With that being said, our satellite actually has a different resolution size of 30 by 30 meters per resolution space, which means roughly 2 to 4 of these 1x1 meter plots will fall in the satellite light band space, and if you do the math, that ends up not representing much of the total space we are trying to predict. Our total training set ends up representing less than 1% of our total marsh plots, which adds some uncertainty to our predictions. This brings us to our methodology, the random forest model. The model makes multiple decision trees that are randomly created using the independent variables given. It selects the best probable result given each tree's level of prediction of a certain class averaged together, which would be the pink bar in this case. In actuality, there are hundreds of these decision trees produced in the model, far more complex than the ones we are showing here. This is in order to prevent overfitting so that we are less likely to misidentifying the same object that just barely differs in its light reflectance band from the resolution space. This is one of the 500 trees generated from our Phragmites classifier for reference. If we zoom in on a small area, you can get an idea of how the model differentiates the different scores in light reflectance data. After running the model, we found that while the random forest algorithm struggled to predict ordinal scores for a species, it could identify whether a species was present or absent from a spatial resolution with only 15 to 20% error. This is our final prediction for August of 2016 for the abundance of Phragmites, the invasive species previously mentioned. The left side of the visual shows our original dataset, and the right is our model's predictions. And here is the same visual but for Chenoplectus americanus, a species that the McLaughlin lab is currently interested in. As you can see, both models successfully predicted, with some noise, the relative species abundance in the marshland. Next, we plan to validate the model on past years and, if accurate, make predictions about past populations to see how they correlate with changing environmental factors like the rise in CO2 and the growth in the Phragmites population to complement work already completed. This concludes my summer research project. Thank you for your time, and I hope you enjoyed this talk.